Share screen. There we go. All right. So this is Enterobacteriaceae part 2. All right. So we had part 1 Thursday, and it was a lot of the biochemical reactions that we got given, and we had it all back on the back sheet. So we have all this info, and then today in lab we're going to go back to API. So we have what's known as API E, 20E for the enterics or the Enterobacteriaceae, and we have 3 organisms plated, so they're smelling really, really good, and that's the way you know you're in the enterics, is because they smell really, really good. So we have 3, and you'll have 2 to choose. You'll have all 3 on your table, and then you'll choose one of the 3 to ID. And I just ask that you switch with your partner, that y'all don't use the same one. Just use one. Pick something different out of the 3. Pick 1, 2, or 3, and set that up for the API. So that's basically, we've got some tests that we'll look at and do on the organism itself today. But the real key is to put your eyes and your brain on the plate, because what I did was I set up a blood and a McConkey. And for most of these organisms, most of these are going to come out of the urinary tract. So when we say that we have a urine culture, okay, urine culture is basically on blood agar and McConkey. That's the 2, that's it. You don't get any more than that. Routinely, you're putting urines on those 2 plates. So this is your good, your best, one of your, you're going to get them again next week, but this is your one chance to see what I got for you today, which is E. coli, Klebsiella, and Proteus. Okay, so this is your chance to get a look, smell, look at the colonies, look how they look on the blood agar versus how they look on the McConkey. What is different? What is the same? Can I see that colony? If I saw it again, would I be able to recognize it? So this is one of the big ones that you'll get your hands on. Some of the most common organisms you'll see in practice. So these 3. Okay, next week we're going to come in with some of the Salmonella and Shigella. You're not going to see those daily. Okay, those are going to be one of those. Hey, I got a stool culture. We're screening it for Salmonella Shigella. Okay, so those are going to be a little more rare in occurrence, but you'll definitely get the textbook of what's going on. We've got some media that we're going to use called heptoin, and we'll definitely set up the Salmonella Shigella on that. Okay, but today this is urinalysis, urine culture 101. You're looking at 3 of the big ones you'll get more often than not on these 3 from UTI. So take advantage of that, whether you got to take pictures or whatever you want to do. I'll allow the phone to come in and take a picture, so you can have that to study back, to have your own picture. So once you take your picture without your gloves, right, put your phone back. So open it with gloves or take a picture through the, but we can do that, and you can set up for these 3 and take a picture of these 3. Like put the 2 plates out there, take a snap, say that's E. coli, take a snap, say that's Klebsiella pneumonia, say that's Proteus mirabilis, I think is what I put out, mirabilis. Okay, deal? All right, so we'll do that downstairs today. So we got some fun going on downstairs. It is a little smelly, but right now it's captured in the incubator, but we're going to open that incubator up when we get going. Okay, so good thing we're wearing the mask, because this is one of the labs where students will ask, can I wear a mask during this one, because it is kind of smelly. Members of the family Enterobacteriaceae quickly colonize, and they grow in the GI tract of humans and other animals. So that's where these are located. All right, these are not in your urinary tract to start off with. Okay, they're going to find their way there. The majority of these are non-pathogenic. They compete for space and nutrients to prevent pathogens from gaining a foothold in the GI tract, causing GI distress. So these are kind of what you would call normal flora. Okay, some of these will be, and we need them. Your GI tract is full of E. coli, right? But we're keeping, the flora keeps other pathogens from finding hold. There are a few species which are always considered to be pathogens, such as they're reportable. Okay, so we got some reportables we'll talk about today. And in some cases, strains of species considered to be non-pathogens, but are able to cause GI symptoms. So we'll play with that idea of what is a pathogen? Does it make me sick? What is a non-pathogen? It's part of my flora and it shouldn't make me sick. Even non-pathogens can wreak havoc if they establish themselves somewhere else. And that's the key to what we're doing today in lab. Is they were in the GI tract, just minding their own business, doing their thing, and weren't causing us any problems. And then all of a sudden they end up in the urogenital tract. They can go to the central nervous system, respiratory. That's when we end up having problems, when they get set up in a different spot. Keep that kind of micro 101, but it is important that you understand that as we move forward. So we have Citrobacter. That's the one we're going to start with. And I think if we look. So this is all out of chapter 19. Okay, so you got a list of these. You've got the media they grow on, selective differential, their nutritional requirements. But you can look at the colony appearance and characteristics on page 345. We see that. So you got a Citrobacter species, and it says we do McConkey. Okay. So it's late lactose fermenting, therefore it's a non-lactose fermenter after 24 hours. It lacto ferments after 48 hours, so the colonies are light pink after 48 hours. So McConkey could help us with Citrobacter. On Hectoin, it's colorless. Okay, and then who knows what XLD? Did y'all talk about media at all in intro micro? Did y'all go through the medias? Like what? The main ones though? Did y'all go over the stool ones? Like what do we use for stool? No? Okay, it's okay. All right, so XLD stands for xylose lysine deoxycholate agar, XLD. Okay, I like to call that our Christmas tree. Okay, because sometimes we'll use that as the red and versus the green. So you see that sometimes in a biplate, and I call it Christmas because I associate red and green with Christmas, but that's just the way I remember it. So Citrobacter is an opportunistic microbe which is able to cause disease when the host resistance is lowered by either disease or drugs. So if you're on immunosuppressants or you have an illness that's causing you to be immunosuppressive, Citrobacter could be a problem. Okay, otherwise it's really, it's going to have to be somewhere else. So it goes somewhere else. It's associated with urinary and respiratory tract infections. Okay, so how does a urinary tract infection present itself? Who's had one? Raise your hand. What does it do? What is the presentation? Our patient's going to tell us what? It burns on urination. I have lower abdominal pain, maybe back pain. So we have those. Maybe I'm running a fever. So we see it quite a bit. But pain on urination is really the key. We'll hear that a lot with something going on with the urinary tract. What about respiratory tract infections? How do they present? Like in a couple of months, like in these 1st of November, y'all come in and I think you have either an upper respiratory distress or you're in lower respiratory, what am I going to hear from you doing? Yeah, you're going to be coughing, right? Okay, so coughing and hacking. So if it goes to pneumonia, which that would be lower respiratory, right? That's in the lungs. And we'd see that's why you get a chest x-ray when you roll into the ER. I mean, you're just going to get one of those, kind of like a CBC for us. Get a CBC and a chest x-ray every time you come in. But we'll see respiratory tract infections could be Citrobacter. So what are we getting out of this lecture? We're getting introductions to our Enterobacteriaceae. And these are names that may not be familiar to you, like this one, Edwardsiella tarda. Anybody heard of that before today, or you've heard of that one? Good. Did we have it somewhere else already? Did we? It is the only human pathogen in the genus Edwardsiella. So the tarda is the one that we're focused on with that. It's normally in reptiles and freshwater fish, and the infection manifests itself for humans in GI disease, gastrointestinal. Okay, so you can imagine how we would get that eating fish, maybe with reptiles, could be. Enterobacter, so those are kind of our first two, Citrobacter and Edwardsiella. Enterobacter is also considered an opportunistic. We have Enterobacter cloacae, which is commonly causes urinary and respiratory tract infection, so a very common theme going right now, right? And here's the big one, Escherichia coli. So again, this is, you know, we always like to say, well, I would say probably number one on the charts, as you would say in the micro lab. Who have we seen the most of in our career? I think everybody would tell you this is it, E. coli. Well, look at this. So E. coli is associated with a wide variety of disease states. We got pneumonia, we got septicemia, we got diarrhea, gallbladder, meningitis, pyelonephritis, hylitis, peritonitis, gastroenteritis, appendicitis, endocarditis, cystitis, you name it. All right, it could be E. coli, could be the cause of all those. The one that we worry most with neonates is E. coli K1, and it's the meningitis causer for neonates. So how does a neonate get E. coli? And this, I'll give you one of my little side theories about something. What do you think? Neonates and E. coli? All right. What's our source? What's our source? Where's the neonate going to be picking up E. coli from?
Could it be the mother?
Could be the mother. Could be themselves, right? Poopy diapers, right? Poopy diapers, babies, infants, right? Infants really, and this is one of the many that I've heard in the last about sudden death, infant sudden death syndrome, is that somehow, some way, there's an E. coli toxin in that crib, the baby's getting exposed, and it actually causes kind of like a suppression of the respiratory while they're sleeping, and that's in that get. So we really don't know the mechanism behind infant death, sudden infant death syndrome, but that is one of the theories, is the E. coli. Okay.
Was it from not changing like the diaper often enough, or like the baby?
Well, just that you just don't clean the sheets, you don't change the bedding, you're putting down for naps in places that could be contaminated with E. coli from like daycare settings and other kids and that kind of stuff. So yeah, it's never a happy day when that happens. So here's our E. coli. From our sheet, did y'all bring your sheet today? What do we got for E. coli on our little sheet guide, our little guide to many, right? We're on a backside today because we're on the gram-negative rods, right? And you see that we have lactose fermenting. We go over to pink on McConkey, and we see Klebsiella over there. We see E. coli over there. We see Enterobacter over there. We see Citrobacter over there. So we got all those gram-negative rods over on the left. We have our non-lactose fermenters, which are colorless. And then we see an oxidase, and we're going to get to those, I think, coming up next lecture. So we'll talk about those next lecture. But you see oxidase negative, lactose negative, oxidase negative, we're going to see on next week's lab, Shigella, Salmonella, and we see Proteus. So you're going to see today, you're going to see a couple of gram-negative lactose fermenters. You're going to see one that is a lactose non-fermenter. So you're going to want to see that on the McConkey too. All right, so this is where we are. So gram-negative, rods, lactose fermenters are key. McConkey's our key media there to differentiate those select. Because this is an electron microgram of a gram-negative rod. The good thing about urines is when you do urines and you look at them when they're infected, we do a microscopic review. You can pretty much tell when you've got an E. coli load, okay? Because when you look at the urine, even though it's colorless under the microscope for the urine, you can tell they're rods. You don't know they're gram-negative rods, but you're hoping they're gram-negative rods, because if we get a rod, we want it to be gram-negative. We can deal with that. Gram-positive rod's a different story. Okay, so you'll see the elongated rod in the urine, right? Not even stained, just under the microscope when you look at somebody's urine that's infected. It makes it easy for us when we see that. So this is the gram stain, which we'll be doing today. Yay. Y'all love gram stain? Okay, I think it helps. I think it helps put the connection. So we definitely will be looking at gram-negative rods today under the gram stain. So that's what you should get. Hopefully you'll get that today. I think you will. Should be pretty easy to pull off. And I like putting the gram stain in on the setup day because we just did the setup. Take y'all, 30 minutes and y'all be done, right? I'm gonna gotta give you something. So here we are on that looks like, that could be blood agar, bad picture. I don't think it's chocolate, but you just see these colonies forming on the agar. This looks like a McConkey. This looks like a lactose fermenting, picking up the pink. So you definitely see that. That looks like some of our agar we had last week, where you had a split in it, or somebody took their loop and kind of got it in there. I don't know whose that was. I remember seeing one. Might have been mine. So this is the hectoin enteric agar. Now we could do that today. We had some hectoin, but hectoin's really, for us in E. coli, what happens is all the ones that we don't care about, all the other gram negatives that we don't care about, will have a different color on hectoin. Okay, there's going to be a real key to Salmonella and Shigella on hectoin. We'll cover that. We'll cover it today when we get to Salmonella and Shigella, but we're going to talk a little bit about it next week when we actually break out the hectoin enteric agar and put these on them. So here's the key. Here's why we're number one on the charts for E. coli. More than 80% of UTIs are caused by E. coli. Pretty much every UTI that happens in the hospital is a UTI from E. coli. But only a few strains are responsible for the most of the serious infections. So we have one type of E. coli called E. coli ST131. This particular app that's traveling from the bladder to the blood, not good, and kills thousands of people in the United States every year. So what you're going to see is E. coli gets a little more variety, like it's strain 131, but we'll have some abbreviations that we key in on. But the first one you'll be introduced to is ST131, kind of in your not UTI, your FYI, which is we need to put that in our memory bank because we definitely don't want a urinary tract infection, which would be a bladder or, leading to the bladder that would lead to the kidneys. Well, this one just jumps straight from the bladder to the blood. That's the septicemia part, okay? E. coli causes 30% of the nosocomial UTIs. That's hospital-acquired UTIs. And those others that could cause in that group are saprophyticus, and we got that S up there. We have S. aureus, and Enterococcus, Proteus. We got S. albicans, or C. albicans, sorry, not S. So who wants to help me with my S's here, my C? Is that saprophyticus? Is that like strep saprophyticus or staphylococcus saprophyticus? We already had our staph and strep lectures, right? Did y'all list out all the organisms that we had to look at? Are y'all making a data bank, or are you listing organisms as you come across them? Be a good way to study when you come down to the final. So what do you think? Think it's staph or strep? For saprophyticus. Look at your, think of this. Maybe this will help you. Flip that baby back over and get over on the staph side. It's under staph epi, so I think it's going to go staph saprophyticus. All right, there's hemolyticus right under that. So it is a staphylococcus saprophyticus. Staphylococcus aureus, so I think y'all would have known that one, but I wasn't sure because sometimes abbreviations can be confusing. And what about C. albicans? What is that? What is C. albicans? I don't know if it's on this or not. I don't think it's on.
Is that your Clostridium?
Clostridium albicans? No. Now think of UTIs. It's not always bacteria that causes UTIs. It could be something else. Think of normal flora. Sometimes people have more of this than others. Have problems with this more than others, especially on the female side of things. That didn't ring a bell. That didn't give a good hint. Female side of things. Kind of associated with yeast, right? Yeast infection. Candida albicans. Okay. We ain't got there yet. It's coming. It's late in the semester when we get over to the yeast. Just seeing where we were. E. coli can potentially be a deadly bacteria. That is, it can cause abdominal cramping, dehydration, bloody diarrhea, and that's one we're keying in on because that's one of our big E. coli's we look at, and there is a particular way we test for that one. And for up to 8 days after exposure, most people recover within a week. Some may develop a hemolytic uremic syndrome, which is a type of kidney failure that gives us easy bruising, an unhealthy pale appearance, and a decreased urine output. So basically what that E. coli has done is gotten to the kidneys. There are distinct serotypes of E. coli that are associated with gastroenteritis, and they are classified according to their mechanism in which they produce pathology, so the disease state, okay? Is anybody familiar with that one? That big E. coli that I'm talking about? Heard anything about a bad E. coli? All right. So the first one we'll talk about, these are the bad ones that cause the state. So we're classifying this as an enterotoxigenic E. coli, also known as traveler's diarrhea. Anybody been to Mexico? No. Lately? No. You ever heard of traveler's diarrhea? Don't drink the water because it's contaminated with fecal material, and you drink it and you get diarrhea. No? Nobody's warned. Who's been to Mexico? Who's been to anywhere in Mexico? What did they tell you not to do? Do not drink the water, right? Don't even drink the ice in your drink, right? Bottle, bottle, bottle. Everything needs to be bottled. Everything needs to be UV ultravioleted and cleaned up, right? Because traveler's diarrhea, that's what we see is we go and we show up on a trip and we're like, this is so fun, this is so great. And we end up getting a fecal contaminated water, okay? And we end up with enterotoxigenic E. coli, which is causing us traveler's diarrhea. So we have diarrhea the whole time we're there. Not a fun trip. So we get fevers and chills and headaches and vomiting, incubation 1 to 2 days, lasting a week, about your vacation, right? And you've transmitted it by eating contaminated food and water. So it actually lasts like a cholera-like illness. So you get this just watery diarrhea, like you came across a cholera, okay? Not a fun one, but that is our enterotoxigenic E. coli. Pathology is associated with an enterotoxic. So enterotoxigenic, enterotoxin is produced, all right, from that E. coli that's causing the diarrhea. And basically, y'all know how diarrhea happens? Y'all know what causes that? So anything that irritates the lining of the intestine to the point where fluid enters, okay? So any damage or irritation to the epithelial lining, there is now water that is coming out of that tissue into the GI tract, causes this watery diarrhea. When we isolate it, the colonies of enterotoxigenic E. coli, that's ETEC short for that, it's indistinguishable from normal intestinal E. coli, and it must be identified based on the toxin. So if we did a culture of that diarrhea, we would probably just find E. coli like we normally would from a stool culture. We wouldn't be really panicky about it. So we definitely want to check for the toxin. Okay, so that's one. That's our enterotoxigenic E. coli. Number 2 is our enteroinvasive E. coli. And this is similar to shigellosis infection, where we get fever, cramps, and blood, mucus in the stool. So a bloody, mucousy stool gets transported, I guess you would say, to the lab. And it's from contaminated food. Okay, so we talked about how does food get contaminated with E. coli, right? Anybody? We could think, oh, well, it's the food prepper, right? We could say that. I mean, it could even be some of these are linked to what? The field where they were growing. Like let animals in the field. The animals had pooped on our food, right? And then they didn't get washed off. So if you ever eat anything right out of the bag, you could end up with enteroinvasive E. coli. Could, because then it was contaminated. And sometimes we can recall those, but probably not after a few people get sick. And then they get a little epidemiology and trace it back to a contaminated food source. And you'll hear like, hey, don't eat the spinach at Subway. Or don't eat the spinach from Kroger. Wherever the source was, when they finally figure it out, they'll trace it back and say, you know, if you had bought that, if you hadn't eaten it yet, throw it away. So what happens is the pathology is associated with penetration of the mucous membranes. So these strains are biochemically and serologically similar to Shigella, which we learned from biochemically. We learned what about Shigella on last Thursday? What did we learn? Biochemically, Shigella is inert, right? Meaning it doesn't show any reactions through the biochemical. All right. So you could associate the enteroinvasive to Shigella. Enteropathogenic E. coli, EPEC. So all these little abbreviations, EPEC. We had EEC, and we had what was the other? ETEC. So we got ETEC, EEC, and now we got EPEC, which is enteropathogenic. It's associated with hospital-acquired infantile diarrhea. Adults do not have this problem, but medical personnel can be carriers and transmit it to newborns. And all of a sudden, your newborn is having diarrhea with blood and mucus in it. And I don't know about you guys, but if I'm having something and it's bloody and mucusy, right? It's probably going to get your attention. Definitely if it's bloody. It is a real panic button in the hospital, especially when a patient starts having bloody diarrhea. Because the first thing in the hospital you think about is what? GI bleed, setting us up for getting blood ready. So you might get that call. Hey, we just had a patient, got a bloody stool, got blood in their bed, got blood in the toilet. We need type and cross 2 units of blood just in case, right? You might get that connection. So that's EPEC. EPEC, the method of pathogenesis is unknown, but because strains are not invasive, nor do the strains produce toxins. So no toxin here with enteropath. Isolates do possess a chromosomal gene which codes for adherence factors allowing organisms to bind tightly to the mucosal cells of the small intestine. And I just told you when that happens, when it gets to the mucosal lining, that's where the diarrhea, that's where the blood, that's where the mucus is all generated from. Right? It's irritating in that mucosal lining of the GI tract. This is the one I was hoping you would know. I was hoping that somebody along the lines, sometime in your career so far, I know it's been short, but E. coli 0157 is the E. coli that we really, really hone for, we look for, screen for, especially if they're having diarrhea over a period of time. Anytime we're throwing it into a Salmonella Shigella, usually now E. coli 0157 gets put in there. So we know it as pus, hemolytic uremic syndrome. So this is associated with profuse bleeding and kidney failure. So hemorrhagic colitis, this is bloody diarrhea. So this is, we're looking at blood in the stool. There's no GI bleed, but this is into the stool and we see blood. So the first thing we do with stool is do what? We do a hemocult on it. We screen it for blood. The nurses will do this. They will take it. They will screen it for blood. They will say we have blood in the stool. Doctor wants a stool culture. The doctor wants to look for these pathogens and see if that's what's causing the diarrhea. Severe abdominal pain and grossly bloody diarrhea. There is a cytotoxin that's produced, and these strains are referred to as STEC, S-T-E-C, Shiga toxin producing E. coli. We got all these abbreviations, and you know what abbreviations are fun for. Text, right? A lot of abbreviations. So Shiga toxin producing E. coli. How are we going to ID it? This is key. We're going to look for it because this E. coli is a non-fermenter on sorbitol McConkey, the SMAC plate. Sorbitol McConkey, S-M-A-C. Okay? So, in your micro lab, you should, say if we say we got the blood and the McConkey down there today, and what if we suspected we had an E. coli with a bloody diarrhea and we want to screen it through this? Maybe the diagnosis is bloody diarrhea. Here it is. Here's your stool culture. An E. coli would usually get ignored on a stool culture unless it was 0157. So you would actually need a McConkey plate with sorbitol in there. Okay, if we set that E. coli up on that plate and it doesn't ferment sorbitol, that means it stays clear, doesn't pick it up, didn't change, then we may have an E. coli 0157. That's our screening growth, but then we also will look for the Shiga toxin. And we can do it by detecting using inoculated gram-negative broth or from a liquid stool. Okay, so the Shiga toxin being produced would be the other thing we could test for. And if we suspect it, we take all those stools with acute community-acquired diarrhea, should be cultured for STECO 157. So community-acquired diarrhea, what does that mean to you? Patients with acute community-acquired means they came in with it, right? They didn't have the diarrhea once they got in the hospital, because we associate that with maybe antibiotics being used, and maybe that's C. diff. We'll get to him later. These stools should be simultaneously assayed for non-0157 STEC, Shiga toxin E. coli, with tests that detect Shiga toxins or the genes encoding these toxins. Okay, so we actually need to remember from this one, we grow it on sorbitol McConkey, looking for a non-sorbitol fermenter. So if it's regular E. coli, it will what? Ferment sorbitol. If it's 0157, it doesn't ferment sorbitol, but we can also check for Shiga toxins. So put those two together with E. coli 0157. Clinical lab should report and send that E. coli 0157 isolate and the Shiga toxin positive samples to state and local public health labs as soon as possible. Because what they're going to want to do is they're going to want to type it, see which one it is. And they'll do that genetically. Remember, we got the specimen or enriched broth in which the Shiga toxin or the Shiga toxin E. coli are detected, but from which 107 STEC are not recovered should be forwarded as soon as possible to the state and local laboratory, so that non-STEC can be isolated. So you're screening it both ways. Questions on that? So we have that. We don't have 0157 downstairs. We just have regular E. coli. And we also have this species, Klebsiella, downstairs, growing for you today. And we actually have Klebsiella pneumonia, which accounts for 95% of the isolates from Klebsiella genus. Okay, there was no. I could have done Oxytocca. All right, I could have gone with Aerogenus, but we went with Klebsiella pneumonia because that is 95% of the isolates of Klebsiella that you find in the micro lab are going to be Klebsiella pneumonia. Most often the cause of pneumonia and urinary tract infections. Maybe how it got its name, but also urinary tract infection. And virulence is associated with the anti-phagocytic polysaccharide capsule, and that's the key to see today. Look for that gooey capsule on top of this one. It's unique. E. coli won't have it. Proteus won't have it. Klebsiella will. Okay, so it has the anti-phagocytic, or if you want to say avoiding the immune system, that's what Klebsiella has. A lot of words on this slide. Good story. So hypervirulent. Hypervirulent. That sounds serious, doesn't it? Hypervirulent. Klebpneumon, a new pathogen, infects completely healthy people and can cause blindness. Okay, that's fun to think about. Flesh-eating infections, brain abscesses, and death in just a few days. Presently, there's no commercially available test to accurately distinguish classical and hypervirulent. Both strains of Klebpneumon can be deadly, but the classical strain is more likely to infect patients with underlying disease who are immunocompromised and hospitalized. By contrast, this hypervirulent strain can infect healthy, young community, causing sudden life-threatening complications. This hypervirulence Klebsiella pneumonia is largely due to genes located at the large virulence plasmid, a possible target of NAT. So I don't know if I would, with all that being said on the slide, are you worried about it? No? Nobody's worried about it? While the assumption is that this pathogen spreads from person to person through food and water, the mode of transmission is still unknown. So far, it's been described most often in Asia-Pacific Rim. So I'm going to get help from our students here. What is the Asian Pacific Rim? Adam, help me out. You have no idea? So that's not a worldwide definition. What does Asian Pacific Rim mean to everybody else? We'll go with you. Asian Pacific Rim.
This is an English term, so
It's an English
I
term.
can't
So,
help you with that. So.
well, let's ask one of our English students then. Mary's always good with answers. Mary, what is Asian Pacific Rim? Where would you think this would be located in the world?
Like Pacific Ocean. Something.
Pacific Ocean, that's a big place, yeah. So over toward Asia in the Pacific Ocean. So we're talking all the way to Japan? We're talking all the way to China. Where are we going?
I
Tokyo.
don't understand geography.
You don't understand geography. So this means, basically what this is saying, what I'm gathering is this means nothing to y'all, right? Even from our Chinese students, our Saudi Arabia student, to our English, no, nobody knows. India, we'll try India. No yet? No? Okay. All right. In North America and Europe, where there is a low prevalence of hypervirulent strain, many clinicians are unaware of this strain of Cleb pneumo. There's no diagnostic test, clinical lab. So just take this for example. Let's take this for example, right? All right, so you're reading this, right? So what happens if we have an outbreak of this highly virulent Cleb tomorrow? What happens? We have no test. We can grow Klebsiella, all right? We can check for it. Sure, it's going to ID as Klebsiella when we do it. What would happen? I know we joke about this kind of thing, but this is kind of how we were really what? How we kind of got COVID. You want to think about it, right? We watched this. We had students in the virology lab, and we sat and we watched from day one how COVID was coming, right? And we watched from day one how all of our health officials, all of our expert scientists, we watched 2020, right? Everything was over there, everything was over here. And we kept saying, well, there's a plane landing in Seattle with 300 Americans on it with possible exposure. Oh, they're going to isolate them in a military base for 2 weeks. There's a cruise ship. Off it goes. How many cruise ships did we have abandoned with people on it? All right? This is kind of the same kind of story. This is kind of the same kind of thing. Yeah, but this is a bacteria, so we're not really worried about it, right? But this is kind of how things happen. This is kind of how they all snowball. So we don't have a test. Why not? Right? Same thing with COVID. Why didn't we have a test? Right? Because tests don't just happen like that. But boy, when we need, like, well, we got to get tested. So think about that when we read something like that. Just think, it could be the same state. We think about it. It's over there, Asia Pacific Rim. Nobody even knows where that is, right? And then all of a sudden, here it comes. And then we have an outbreak, and then we're still dealing with it, right? Anyway, Proteus, Providentia, Morganella. This is our ppm group that we had introduced on Thursday. So the Proteus we have downstairs. So you'll get a good whiff of Proteus today. Nosocomial infections, urinary tract, and wounds. So these are kind of moving into our wound section. We haven't had wounds yet. So Proteus is definitely a player in the wounds that we see, infected wounds. The pathology is they produce urease. Well, we know we've been checking for urease, right? Urease is an enzyme which hydrolyzes urea into ammonia and CO2. So we have that down here. don't see CO2 down there anywhere. Urease is an organism, enzyme which hydrolyzes. That's the hydrolyze. That's urea into water and produce ammonia. Is that ammonia down there? Is that right? Okay. We did have that. It raises the pH of the urine from acidic to neutral, which favors who? The bacteria. So this, to be a UTI, makes sense because, why? It actually changes the pH of the urine. So urine is acidic, right? It knocks out the growth of bacteria. They don't like to grow in an acidic environment. Well, this one's figured out how to change that environment. It's by producing urease, the enzyme that changes ammonia into ammonium hydroxide. That's where I was trying to say. Ammonia, this is ammonia, right? Combines with water to produce ammonia hydroxide. Okay, sorry, I was looking at it that way. It didn't make sense. There it is. There's Proteus mirabilis. So we have that one growing on blood agar and McConkey downstairs. The key here is what we're going to look for is we're going to see this swarming on blood. But my question when you get down there is look at it on McConkey. Okay? And then I'm going to ask you downstairs if that's swarming on McConkey or not. Oh, there it is on McConkey. Is that swarming? No, right? So you're going to see Proteus not swarming on McConkey, but swarming on blood. And as you can see, who do you think this is? Who do you think this is? Well, that looks like McConkey both. Both are McConkey, right? And McConkey, when it's lactose fermenting, is what? Picks up the pink. This would be a non-lactose fermenter on McConkey. You want to check it? Let's look. Let's see if we can find Proteus on here. Oh, we get indole, but we don't really get anything else. It says it has a chocolate cake odor. We'll see. We'll see if it smells like chocolate cake down there today. Y'all tell me. You'll tell me when y'all get in there if it smells like chocolate cake. The chocolate cake would smell good. All right, so one thing we do see and what we are going to do some testing on is indole. We do have an indole developer. We're going to give you a little piece of filter paper and we're going to have you put some these organisms on there and then put a drop of indole developer on on all of these. So we'll get that. So we'll see who's indole, who's not. If you look, you can already see that that's one of the big tests we like to do. Because what you're going to see is that we have Klebsiella, which is indole negative. We got Proteus mirabilis, which is indole negative. And we have E. coli, who is indole positive. Okay, so that's kind of what we're going to work on today. Serratia marcescens. No, I saw him over here. That's on the gram-negative rod, lactose fermenter. Urinary and respiratory tract infections. Should have that nailed down by now. Hospital acquired. Patient subjected to instrumentation. Maybe a catheter. Maybe a trachea. Maybe an IV. Maybe respiratory equipment. Or even maybe patients on antimicrobial therapy. Yay, right? Don't see anything up there unusual. Maybe the trach, maybe if you're not in the ICU on somebody with a trach. IVs, really? IV can introduce a microorganism? Y'all have all been phlebotomy, right? So keep that in mind. Virulence is associated with multiple resistance to antibiotics. So the more resistant an organism is, the more virulence it has. Okay, so antibiotics given over time may be set up for Serratia marcescens to be resistant, more virulent, not going to be able to take care of it quickly, but look at the things it could be introduced with. Catheter, IV, and then the patient that's on antimicrobial therapy. There's a lot on this, right? Has anybody counted them up so far? What are we up to? Don't know. A lot. Yersinia intercalyptica. Yersinia. I used to, when I was doing stools, we always called them, we always cleared them for Salmonella, Shigella, and Yersinia. That was our three. They took Yersinia off. They said, oh, you just got to clear them for Salmonella and Shigella. What is Yersinia? Infants and young children, enterocolitis and diarrhea. Sounds like I should still be screening for it. Older children and adults, it shows as lymphadenitis and ileitis, clinically resembling appendicitis. Reservoirs, domestic animals. So Yersinia intercalyptica. Transmission, contaminated food and milk products. And then we see this Cefsolodin, ergocin, novabiacin. That sounds like sin auger, C-I-N. And that's what? What do these sound like? Novabiacin. Sounds like some, kind of sounds like our modified Thayer Martin a little bit, right? Remember that one? For Neisseria, right? So this is a sin auger, and it is for Yersinia intercalyptica. So it only will do what? Selective and differential, meaning it will grow on that, and others will not. To me, that looks like, whoops, what did I do? That looks like blood. That looks like McConkey. There's the sin auger, and you see growing, picking up Yersinia. This is probably the more famous of the Yersinias. This is Yersinia pestis. And we know this as the what? The Black Plague. The etiological agent of the Black Plague during the 14th century, bubonic plague, was responsible for 25 million deaths. From 46 to 53 of 1300s. It disseminated about a quarter of the third of the population of Europe. It was gone. The Black Plague exists in 3 clinical syndromes, all of which characterized with high fever and malaise. Shanghai, 1936. This is Alexander Yersin. Now you might know where Yersinia picked up its name. Plague studies in 1965, showing you no gloves, right? 1965. They at least have lab coats on. Here are the 3 clinicals. We have bubonic, we have pneumonic, and we have septicemic. So these are the 3 different ways Yersinia pestis, or the plague, presented itself. Bubonic, inflammation, swelling of the lymph nodes, mainly between your legs in the growing area or under your arms. 75% mortality if untreated. Pneumonic, transmitted person to person through respiratory droplets. Rise after the initial fever, mortality 90% if untreated. And septicemic, during this phase, the organism invades the blood, following the bubonic or the pneumonic plague. So we had 2 different stages, 3 different clinical syndromes with Yersinia pestis. That's the bubonic. It's hard to tell. See a swollen leg. This is the pneumonic. That looks like gram-negative rod in here. It's like red cells, plague victim. Yersinia pestis on blood. Chocolate. Okay, so where does it come from? It comes from rodents. Rats, mice, ground squirrels, prairie dogs, and rabbits. It's usually a flea that leaves those reservoirs with it. Okay, so the reservoir is not killing the rat, the mouse, or the ground squirrel, the prairie dog. So you can get transmission from rodent to rodent, but rodent to man. But the vector is the rat flea, the Xenophilus cheapus. It develops after a bite of an infected flea, direct contact with an infected animal, okay, inhalation of contaminated aerosols, and once it's in, it's phagocytized by the neutrophils, but can multiply inside those neutrophils. Robust production of capsule at 37 degrees, allows many organisms to escape into the lymphatics and the lymph nodes. There's our black rat. He's setting a trap. There's our flea. A flea. Flea. Flea and flea larva. Looks like the rat eaten by fleas, maybe. All right. Oh, that's a ground squirrel. Sorry, I didn't read my own slide. Ground squirrel. Flea. Infected flea. Prairie dog. Animal trap. Okay. So yes, there is, I think there's a question I know in the question bank that it's one of those questions that kind of hang on and gets sent through. Like it's in this group, but it's been corrected once I upload it over here, but then I got to upload it into another Blackboard shell. So I uploaded over here and I forgot the correction needed to be made. This is the one the Yersinia pestis is the one everybody texts me about. So I know it's almost like a fun game when I upload the questions. I always forget and somebody texts me and goes, I don't think this is the answer. It gave us another Yersinia instead of pestis for somebody kicking a groundhog or prairie dog and getting a flea bite, right? So we connected there. And I think it always starts with a 32-year-old man in Arizona. So if you see that question as you're studying for this test, you'll know it's from. Needs to be. If it hasn't been changed, text me and say, this one you forgot to change that answer again. I'll try to remember this time. Shigella. Shigella species are genetically identical to E. coli. In fact, they're serotypes of E. coli. So we talk about Shigella as this, sidebar. We also need to connect it back with E. coli. Taxonomical change, this instance was rejected due to the clinical importance of keeping the name Shigella. So there was, with microbiologists, sometimes we like to change the names. And sometimes we can't get them changed. So they didn't want to change Shigella to E. coli because Shigella has this known, hey, it's serious. Everybody recognizes Shigella as a pathogenic diarrhea causer. And they didn't want to go back and just say it's E. coli. It causes bacillary dysentery. Okay, so we associate Shigella with dysentery, fever, cramping, abdominal pains, diarrhea with blood and mucus. Kind of sounds like our E. coli 0157. Spread favored by poverty, overcrowding, and poor sanitation and hygiene. Transmission, as few as 10 to 200 organisms may cause disease from contaminated food and water. So think about a loop full of bacteria, and you just put that on some food, right? That'd be enough. Be enough, right? It said 10 to 20 organisms, or 200 organisms. Shigella with homosexual men, shigellosis can be sexually transmitted. Pathology, invasion of the mucosa leads to ulceration. Most Shigella, more than 90% isolates reported in the U.S. are Shigella Who wants to take a shot at that? We always like to throw this out. I've got one way of saying this, but I want to hear y'all's way.
Dysentery.
No, the Shigella with the species name there. The group D. Oh, the first one's dysentery. I got that one. Dysentery, flexnaria, boidy. What's that last one? How would y'all say that? The EI always gets me. Because if it was IE, I just call it Sony, right? Or is it still Sony? Or would you go Sony? Huh? Still go with Sony? Okay. All right, good. I like that. All right, so that's group A, B, C, and D. You need to know those groups. You need to make sure you've got your Shigellas in their group. So Shigella dysentery is group A. Shigella flexnaria is group B. Shigella boydii is group C. And Shigella soni is group D. I'm not good at these, okay? But if you want to remember it that way, that all day, boys flung, cats blissfully, dogs split. Okay, it was easier for me just to memorize this group, okay, than to do that. But either way you want to, it's a helpful mnemonic if you'd like it. All right. So we're going to see Salmonella and Shigella next week in lab. So next Tuesday when you come to lab, we're going to have Salmonella and Shigella growing for you to see and play with. Salmonella, a little more famous than Shigella. Previously named, their serotypes, Salmonella typhi or Salmonella enteridius. But it's been confirmed that there are only 2 species of Salmonella. We're going to go with Salmonella enterica and Salmonella bongori. All other pre-mutations and nomenclatures are in fact subspecies or serotypes of Salmonella enterica. Therefore, the correct nomenclature for what was formerly known as Salmonella typhi is Salmonella enterica. Serotype typhi, okay? Either way you want to get there, we're going to call it Salmonella enterica typhi, but Salmonella typhi just has kind of like that Shigella. It has a historical sound to it. It has a, we think of typhi as it's just part of it. So keep that in mind. Non-typhoid Salmonella infects both man and animals. So the source of this Salmonella, non-typhoid, is poultry. We know that. Chickens, ducks, and turkeys. Pets, turtles, cats, dogs, and mice. We don't like to talk about guinea pigs and hamsters having this. And other domestic animals such as cows, pigs, sheep, donkeys. Birds, doves and pigeons, parrots, okay, seals, lizards, snakes. Every animal tested has been shown to act as carriers. So if you have those animals in your house, could be a source of Salmonella. If you cook chicken, ducks, and turkeys, could be a source of Salmonella. If you have turtles, you catch turtles. Okay, so you can see quite a bit of sources. Human infections acquired by ingestion of that infected poultry, beef, pork, or water that's been contaminated with feces or urine of infected animals and humans. Plant foods contaminated with urine and feces also transmit the disease. Tools used to slaughter animals may spread the organism from one carcass to many others. So once it's infected, it stays infected until they clean it. Oops. Both Shigella and Salmonella are primary pathogens. They are able to cause disease in healthy individuals and require no predisposing factors. Both are reportable. So if we have Shigella, reportable, Salmonella's reportable. Transmission, contaminated food and water. Prevention, cook your food. Cooking it destroys the organisms by high temperature. We're good to go. There's our little frog. I used to have these, or my kids did. Come in a little, a little square thing, had a little like plant stalk and these little frogs. Definitely had these. Turtle. So here what you're seeing is the rate of reptile-associated salmonella growing in humans. I guess back in the 80s, we decided it was okay to get some animals and bring them to the house. See it increasing. There's turkeys, chickens, more chickens, chicken breast brought at Kroger, Walmart, Aldi. And this is one of those great carriers of it, is the cockroach. Some more isolation, gram-negative rod, growing on like blood or chocolate. Here is the XLD. Let me show you this. I don't think we have any xylose lysine deoxy XLD media, but what you see is look at the colony picking up. Right, the colony changes for salmonella. Its color. So if anything else was growing on these, it wouldn't pick up that distinct color. And I think that's black for salmonella. Okay, so salmonella on hectoin, we're going to see that. We got hectoin. Okay, salmonella shigella auger here, showing the black salmonella, XLD, gram staining salmonella. So we're going to do a gram stain next week on salmonella. We're also going to have hectoin auger to see it growing on that. Salmonella typhi, were we calling it typhi still? What were we calling it? What did we change the name to? Did we change its name? I thought we did. Didn't we? Sorry, didn't we change its name? I thought we changed its name. I thought we were calling it Enterobacteriaceae. Is that all right? I don't know. It's getting late. It's 10 after. Starting to shut down on me. Salmonella, salmonella. Salmonella typhi. Let's look at it. But were we just over here? I don't want, this has happened before. If you're wondering why I'm sitting here batting this around. What did we, oh, that's Shigella. Isn't that what they just said? Didn't they say, hey, we're taking formerly known as Salmonella typhi? We're going to call it Salmonella enterica subspecies, Enterica stereotype typhi. Or is this the non-typhoid? All right, we got to get this straight. So we got 5 minutes to get this straight. What are we going to go with? Zoomland, you can chime in too. So the question is there a difference between Salmonella typhi and non-Salmonella Enterobacteriaceae typhi? Non-typhoid. Oh, this is awesome. What do y'all think? Salmonellas were previously named Salmonella typhi or Salmonella enterica. It's been confirmed that we're going to go from two species, either Salmonella enterica or Salmonella mongori. I didn't know typhi there, right? Formerly known as Salmonella typhi. So what's the difference between non-typhoid and typhoid? That's my question. We'll stop here until y'all answer that.
Is it your reservoirs?
Your reservoirs,
Because typhi is humans only.
human only type is a human reservoir, where non-typhoid is who?
It says both man and animal.
Man and animal. There you go. Very good. All right, that's the difference. So Salmonella typhi, reservoirs only humans, and this is where typhoid fever comes from. So all that earlier was non-typhi. Okay, so typhoid fever, ingestion of food and water. If you get like tomorrow, if you're like, it dawns on you, you're like, I'm still confused, we'll work on it again. But right now, I think we're on the right path to keep these separated. Transmission, ingestion of food or water contaminated with feces, urine of infected individuals. So this is human to human. Chronic carrier. You may have a human as a chronic carrier of salmonella for years. Organism resides in their gallbladder. That means it's going to be shed in their stool, and they can be a source of infection ongoing over and over and over and over. So we have a great story about Typhoid Mary. You can look that up. Okay. So contaminated through leaky water pipe, hypochlorite device. They had typhoid immunizations in Laredo, Texas. Typhoid outbreak in the children's ward. Typhoid fever rose spots. Typhoid cholecystitis. Salmonella lesion of a sickle cell patient. The isolation of Salmonella typhi incubated up to 8 to 14 days. First week recovered from the blood in 90% of the patients. Third to fifth week stools are 85% of the time. We call it a Whittle test, which is a serological test to look for the titer and antibodies to the O and H antigen to the Salmonella typhi. Okay, one of those, was it immunology we talked about this? Whether it was somatic, flagellar. Am I thinking way past immunology there? Remember I thought somewhere lately. Drug of choice is chloramphenicol. The Vi antigen, the V antigen is an anti-phagocytic capsular antigen. So Salmonella is pretty, you can avoid. There's Typhoid Mary. She worked in New York at a food, like a ward, some kind of ward, hospital ward, and she infected, I don't know, 50 cases of typhoid fever, 3 deaths. About 3,000 New Yorkers had been infected probably from Mary. She refused to have her infected gallbladder removed. Immunization against typhi was developed in 1911. There was antibiotic treatment not till 1948. Thus, dangerous source like Mary had been restrained. You had to be, they arrested her. So they arrested Typhoid Mary. Police were met by an uncooperative Mary who eluded them for 5 hours. At the end, she was forced to give samples. Her stool was positive for typhi. Thus, she was transferred to an isolate college on the Upper East Side, near the Bronx, maybe. She unsuccessfully sued the health department. No one ever admitted to explain Mary's significance as being a carrier. So we have a long story here. You've got it if you'd like to read more about Typhoid Mary. After a total of 26 years of confinement, she died in 1938. So they basically put this woman in confinement for her life because she had Salmonella typhi in her gallbladder. Can you imagine that today? I mean, it's bad enough that they're chasing kids down at bars and parties, right? What if they started putting you on, lock you up? COVID positive, they'll lock you up, right? Anyway, we have our other enterics. We'll finish up. Ewingella, Hafnia, Kluvera, Sedecia, Molarella. Those are just some add-ons to the end. Okay, so you got a good story about Typhoid Mary. Now you know who she is, what she has.
I have a question.
Yes.
We're calling typhoid Salmonella enterica. We call non-typhoid the Fungoria.
We're calling Salmonella typhi is the human. Salmonella enterica typhi is the non-typhi.
What about the Fungoria one? We didn't talk about that.
We didn't talk about that one. Not all, yes. Okay, Zoom, we're logging out.
See y'all in lab.